A new chemical probe protects healthy cells from DNA damage, preserving them from one of the 8 hallmarks of aging. The story of this potentially paradigmatic development begins where so much of human health begins, the mitochondria. These organelles are disrespectfully monikered as the powerhouses of the cell, but they do so much more than just provide cellular energy. It's so important, and it even has its own DNA. Mitochondrial DNA, mtDNA, is separate from the DNA housed in the cellular nucleus. While nuclear DNA contains the vast majority of the genetic code, mitochondria carry their own smaller genomes that are essential for cellular functions. mtDNA exists in multiple copies per cell, but when damage occurs, these copies are often degraded rather than repaired. If left unchecked, this degradation can set off a cascade of failures linked to heart condition, neurodegeneration, and chronic inflammation. Published in the German Chemical Society journal Applied Chemistry, International Edition, researchers at UC Riverside developed a chemical probe that binds to damaged sites in the mitochondrial DNA and blocks enzymatic processes that lead to its degradation. There are already pathways in cells that attempt to repair, said Linlin Zhao, UCR Associate Professor of Chemistry, who led the project. But degradation happens more frequently than repair due to the redundancy of mtDNA molecules in mitochondria. Our strategy is to stop the loss before it becomes a problem. The new molecule includes 2 key components, one that recognizes and attaches the damaged DNA, and another that ensures it is delivered specifically to mitochondria, leaving nuclear DNA unaffected. In lab tests as well as studies using living cells, the probe significantly reduced mtDNA loss after lab-induced damage, mimicking exposure to toxic chemicals such as nitrosamines, which are common environmental pollutants found in processed foods, water, and cigarette smoke. In cells treated with the probe molecule, mtDNA levels remain higher, which could be critical for maintaining energy production in vulnerable tissues such as the heart and brain. Mitochondrial DNA loss is increasingly linked to a range of diseases from multi-organ mitochondrial depletion syndrome to chronic inflammatory conditions such as diabetes, Alzheimer's, arthritis, and inflammatory bowel disease. When mtDNA fragments escape from mitochondria into the rest of the cell, they can act as distress signals that activate immune responses. If we can retain the DNA inside the mitochondria, we might be able to prevent those downstream signals that cause inflammation, Zhao said. Importantly, the researchers found that the protected DNA remained functional despite being chemically tagged. We thought adding a bulky chemical might prevent the DNA from working properly, Zhao said, but to our surprise, it was still able to support transcription, the process cells use to turn DNA into RNA and then into proteins. That opens the door for therapeutic applications. The project builds on more than 2 years of research into cellular mechanisms that govern mtDNA processing. While additional studies are needed to explore clinical potential, the new molecule